So my name's Dan, and I lead the applications team at Oxford Nanopore. And one of the things that the applications team does is we like to look for biological questions that that nanopore sequencing technology is well placed to answer. And we're particularly interested in those sorts of biological questions that make use of the unique features of nanopore sequencing. So I'm going to be talking about a couple of examples of that in my talk today, assuming I've got time. But first of all, I just want to say a little bit about the applications team generally. So we're a team of molecular biologists and bioinformaticians, and we're based in a few different locations across the world. So we have a lab here in Singapore, which is run by my colleague, Paula. We're also joined by my colleague from the UK, Justin O'Grady, who runs our translational labs team. And we're joined also by my colleague, Cecil Yule, who runs the genomic applications team, and whose work, what I'm presenting today, is going to be based on. So if you want to hear more about what the applications team does, then please come and find one of us or all of us after this and ask, and we'd love to meet you. So I have two titles. What I'm going to be talking about today, a couple of things, like I said, but I'm going to focus mostly on pharmacogenomics. And if you're not familiar with the idea of pharmacogenomics, it's the study of our genes and how they can affect our ability to metabolize drugs. So we all have a large number of pharmacogenes, and they can do different things, the gene products. So a pharmacogene might code for an enzyme that is involved in the metabolism of a drug, or it might code for a protein that's responsible for the transport of that drug into and out of a cell. So an example of a pharmacogene is the one on the slide, UGT2B17, and I'll come back to this one later on. I can't pronounce what it is that it does, although Lucy can. But suffice it to say that it sticks a small sugar molecule onto steroid hormones at an early part in their metabolism. Now, with this and other pharmacogenes, variants in those genes can affect our ability to perform these tasks efficiently. And so that's why we're interested in studying them. So the Clinical Pharmacogenetics Implementation Consortium has identified 119 pharmacogenes, although different groups would include other genes or maybe fewer genes than that, depending on who they are. But the CPIC has also identified 500 gene-drug pairs as well, which is to say 500 pairs of genes and drugs where the alleles of those different genes and the variants and their impact on the ability to metabolize the drugs is well understood, and it's backed by a robust science. And it could be that a variant of a particular gene doesn't have a huge effect on our ability to metabolize a drug, but in some cases it can be a matter of life and death, and that's why it's really important to understand the genetics. So I'm going to illustrate this by talking about probably the best-known pharmacogene that there is, which is CYP2D6. And this is a member of the cytochrome P450 enzyme family. And what it does is it codes for an enzyme that demethylates drugs. And it's probably one of the reasons that it is so well known is that it's involved in the metabolism of, I think, something like 74 drugs that are in clinical use, including things like opiates, tamoxifen, antidepressants, and so on. But maybe one of the other reasons that it's so well known is that it's one of the most polymorphic genes that there is. There's something like 90 different variant alleles of CYP2D6 that are known, that have been catalogued so far. And these include all the different sorts of variation, SNPs, indels, structural variants, copy number variants, and everything else that I haven't mentioned. And perhaps interestingly about CYP2D6 is that it's got a pseudogene called CYP2D7 that's very nearby, and it can recombine with this and produce other variants as well, which we'll see later on. And as I've said, all of these variants can affect our ability to metabolize these drugs. So the sort of wild-type situation, the normal situation would be if you had two normally functioning alleles, and you'd be described as having the star1 genotype here. And that would mean that you would have the usual ability to metabolize things like codeine or whatever. And the point really being that if you have that genotype, then a physician, a clinician doesn't need to worry too much about the dose that you're being given. It's just the regular sort of dose. But because you can have copy number variation of CYP2D6, it means that you can have more than the regular 2 copies. So if you have 3 copies or more, then you would be classed 3 normal copies, fully functional copies. You'd be classed as an ultra-rapid metabolizer of these drugs. And that can have really serious consequences. And there's an example of such a thing on the bottom. So as I said, CYP2D6 codes for an enzyme that demethylates things. And if you demethylate codeine, then you get morphine. So if you're a breastfeeding mother who is taking codeine, then the levels of morphine can reach potentially lethal levels in the breast milk, and so this is something that you really need to know about. And at the other end of the activity scale, if you're a very poor metabolizer of the drugs, you need to be sure that the level of the drug doesn't reach potentially harmful levels within you. So it's important to understand the genetics behind it. And I'm just going to keep the top sentence on for the next slide. It's not just important to understand what the variants that a person has are. It's important to understand the phasing of these variants if you're going to get a person's optimal drug dosage correct. So throughout the ages, there have been a range of different methods used to look at pharmacogenes, and these include all of the well-known things: Sanger sequencing, quantitative PCR, and so on. And these are good at detecting variants, but not so good at phasing the variants. So although it's useful to know this, it doesn't give you all of the information you need to get somebody's optimal drug dosage. And so naturally, we're interested in slightly more sophisticated methods than this under the circumstances. So there are well-known methods of looking at pharmacogenes that are already commercially available. And so in particular, PCR, there are commercial panels available that will allow you to amplify 40 or so PGX genes all in multiplex. And you also have hybrid capture panels, such as the one by Twist, which is a long fragment capture panel, which again can capture tens of PGX genes. But the problem with any PCR-based method, which both of these are, is that you're kind of limited in the length of fragment that you can, that you can deal with, that you can sequence at the end of it. And that becomes a problem when you're looking at something that's got a complicated gene architecture like CYP2D6. I mean, that's to say that all of these methods are compatible with nanopore sequencing. You just do the library prep that the manufacturer specifies and stick our adapters on, stick it in the nanopore, and Bob's your uncle. But I'm not going to be talking about these methods. I'm going to be talking about something that's maybe a bit more nanopore-y for the rest of the talk. So, I mean, the most obvious way to go about this, to look at people's pharmacogenes, is to do whole genome sequencing. And so what we thought we would do is to start by defining a PGX panel that we wanted to look at. So the good thing about whole genome sequencing is that you're not limited in the target, in the panel size. You can include everything that you want to. So we wanted to make a very inclusive panel. And you just filter bioinformatically. So we started off by taking the 119 genes that the Clinical Pharmacogenetics Implementation Consortium define as the PGX genes. But then we wanted to make sure it was as inclusive, like I say, as possible. So we added in the 173 genes that the FDA recognizes PGX genes. I mean, there's some overlap there, so it's not 173 new genes, but we included all of those. But while we're at it, we can include some blood group genotyping genes as well, and various other actionable targets. So in the end, we've come up with a target panel, a panel size of about 341 genes of unique gene targets, including all of the things that are captured by the Twist pull-down panel, all of the genes that are genotyped by the software tool that we use, which is called FarmCat. And so this is how well it works. So bearing in mind that you don't have to amplify to do nanopore sequencing, you would expect to get nice, even coverage across all of the targets, which hopefully this makes it clear that we do get. So this was done with one sample run on a single PromethION flow cell, and in this run we got an average of, I think, 47-fold coverage, which is probably doing ourselves a bit of a misservice, because I think in the autosomes you'd get more than about 50x, but it's brought down by the fact that it's a male sample, so there's single copies of X and Y here. But what's interesting about this sample, I mean, this is the sample, I think I've got the NA number later on, but the point is that it's the UGT2B17 gene in this individual is deleted on both copies. So this person has what is described as the STAR2 genotype, which means that they can't stick the sugar group onto steroid hormones and metabolize them as part of that pathway. It's not to say that they can't metabolize those steroid hormones at all, but it just means that they can't metabolize them using this pathway. But we can do more than just whole genome sequencing, and so I want to move to talking about a very, very nanopore method that you may have heard about, which is called adaptive sampling. But if not, I'm just going to give a very brief overview of how it works. Because nanopore sequencing takes place in real time, as soon as a strand starts going through a nanopore, then that sequence becomes available to base call. And so within about half a second to a second of sequencing, we have enough sequence from any strand to align it to a reference and to figure out whether we want to carry on sequencing that strand, in which case the whole thing will go through and we'll sequence it as normal. But if it's off target, if it's something that we've defined as being not of interest to us, then we can identify that and we can reverse the electrical potential and boot the strand out of the pore and then capture another one and keep on going and see if we like the next one and so on. And we can by doing this, we can enrich for the desired target regions. Now, an advantage of doing this is that there's no kind of laborious library prep. You just make a regular whole genome sequencing library, which can be done in as little as 10 minutes. Unlike something like a hybrid capture, where you've got to do something like 48 hours worth of hybridization and lots of hands-on steps. But it's also very easy to customize the capture panel that you want. You can add or remove gene targets from your bed file, tell the software that, and it just automatically will then go and capture your target regions. And you can do something else as well. So instead of telling the software regions that you do want to capture, you can, if it suits your experimental design, tell it regions that you don't want to capture and do a depletion method instead. But here we did enrichment. And so this shows the exact same 341 gene panel done with adaptive sampling. Now what we could do is we could run this on a MinION flow cell, and we would get something like the same level of coverage across all our target regions as we would have got if we'd taken a whole genome approach on a PromethION flow cell. But we can also run adaptive sampling on a PromethION flow cell. And we have the option of either getting ourselves loads more data, but we didn't need to because we get enough coverage for all our variant calling anyway, or we can multiplex. And so that's what we've done here. So for this experiment, we took 5 different samples which we barcoded and ran the same adaptive sampling script on. And this is just showing one of them. And hopefully you can see the same one with the UGT 2B17 dropout. So hopefully you can see that the coverage is very even across this as well. And it's also, as the bottom bullet point shows you, even across the different samples, although that's more to do with the library prep, I think, than the adaptive sampling itself. And we can show the two side by side. So on the top, we have the whole genome results for this sample. Underneath, we have the adaptive sampling results for this sample. And I think you'll agree that they look pretty similar. I mean, there's only one thing, I don't want to fall off the back of the stage, but there's one of the 341 where we do get lower coverage than expected, and that's something that we can look into and figure out the reasons for. But I think 340 out of 341 is pretty good going. And when we do adaptive sampling on this run, we got 6.2-fold enrichment of the target regions, which I suppose shows us that we could have done 6.2-fold multiplexing, but we did 5. We got more coverage than we needed, but you know, still good. So let's have a look at some. So you know, we can capture all of the regions that we want to, but let's have a look at some of the variants that we are interested in. So what I'm showing here is these are all 1,000 genome samples, which comes in again later. But this is sample called NA19226. And we're looking at the CYP3A5 locus. So this locus codes for a gene that's involved in the metabolism of malaria, HIV, and TB drugs. And this individual is described as having what you'd call a star1, star6 genotype, which is to say they've got one copy of the regular, fully functional star1 allele, but their other copy is not functional at all. Now, I can't say with any sort of degree of any understanding whether this would affect their ability to metabolize those drugs in any sort of way that would affect the prescription, but it could be that you would need to adjust that person's dose of the drug so that they had the optimal level of drug to do this, to fight the HIV, TB, or whatever the drug was for. But the point that I want to show is that the star6 allele is defined by this SNP, which you can hopefully see very, very cleanly in the IGV plot. But you might be saying, well, you know, this could be cherry-picked data. How do you do on SNPs generally? And so one of the things that we do extensively in the applications team is to benchmark the ability of our technology to call all the different variant types. So in the histogram on the right-hand side, I'm showing our ability, our SNP calling data across a whole genome. And I mean, we're very, very good at calling SNPs. We do this with an F1 value, which is a harmonic mean between precision and recall of 99.8% at 40-fold coverage, which we get more than with the adaptive approach. So basically, if a SNP is there, we'll see it. And it's a similar story for indels. I mean, here we're talking about coding regions, and that's where our indel calling is particularly good. So we get an F1 value, again, this harmonic mean between precision and recall of about 99%. So again, if it's there, we'll find it. But because we can get longer fragments, we can get longer reads, it gives us the ability to resolve more complex genome architectures than just SNPs. So here's an example. This again is CYP2D6. And you need long reads here because of the nearby pseudogene, you need to be able to anchor the reads that you have from 2D6 in the unique sequence at either side. But we can do that. And when we look at this sample, which is NA11832, we can see that this individual has 3 copies of CYP2D6. Now, you might be thinking, that must mean that this person's an ultra-rapid metabolizer. But in fact, that's not the case, because it's only one of their alleles that's functional, and the other 2, the star4 allele and the star68, are not functional. So in fact, they'd be classed as just regular extensive metabolizers, which most of us are, and a physician or a clinician wouldn't have to adjust their dose of any of the drugs that CYP2D6 metabolizes. But again, just to show you that this isn't a one-off, here are some benchmarking results from structural variant calling across the whole genome. So we get an F1 value, this harmonic mean for structural variants of around 96.3% at 40x, 96.1% at 30x. The coverage doesn't really make a huge amount of difference. But that's as good as you're going to get with structural variant calling. It's not because of any sort of weakness on our part. It's to do with the truth set that we use for our comparisons, that not being perfect. So, basically, if a structural variance is there, we will find it. And what we can also do is we can resolve the architecture around the CYP2D6 gene in this individual, so we can see that that functioning star1 allele is on one of their chromosomes, and the star68 and star4 alleles are on their other chromosome. And that we can also see that star68 is a recombinant event between the pseudogene 2D7 and CYP2D6. So, using the long reads, we can get a lot more value out than if we'd just been using a short read approach here. And how do we do overall? Well, what we can do is we can compare the results that we get from our allele calling with the results from the Genetic Testing Reference Materials Consortium program, which is probably better pronounced as GETRM. But the problem is that the database that we have for comparison is a bit outdated. So when we do our allele calling, we can see that we agree with this database in the vast majority of cases, and that's indicated by the white boxes here. But there are some disparities, and these are indicated by a kind of yellowy color. So it could be that we've got it wrong. It could be that the database is out of date. So we wanted to look into this further. Now, because these are all 1,000 genomes samples, it means that we can look at the sequencing results we got, and we can compare them to the right answer, and we can see whether we called it right or not. And in every case here, we did. And from that, we can also decide whether or not, or we can see whether or not the software called the right allele for these cases as well, and it did in every case. So it's that the database that we have is out of date, and our ability to call these alleles, PGX alleles, is in fact very good indeed. Right, in the 2 minutes that I've got left, I'm going to talk about another thing you can do with nanopore sequencing, also with adaptive sampling, so I can stay on topic. But the idea here is that you can use adaptive sampling to do positive enrichment or depletion of target regions. But in the process, you're sequencing every single strand to some degree. And so it means that these are randomly sampled from across the genome. So when you're enriching for a target region, you also get very low coverage across the whole genome. And you can use that for stuff. You can use it to look at things like copy number variants, and the one I want to talk about, which is low-pass genotyping. So with low-pass genotyping, I mean, this is something that I spoke about at London Calling earlier this year, so if you want to hear that in more detail, then please check out the recording of that. But the idea is that with low-pass genotyping, it gives you the ability to call SNPs across the genome, but it's a random sample of SNPs. You don't see all of them. You see some of them, but you don't know which it's going to be any given time. But what you can do is if you have access to a good reference panel, and ideally a long reference panel, a long read reference panel, and I think that that Danny Miller is going to be talking about this in a bit more detail later on, so I don't want to steal his thunder in any way. But what you can do is you can statistically predict the presence of other SNPs that you hadn't seen in your data, but that ought to be there. And what you can then do is you can calculate somebody's risk of developing a particular complex disease. And so just to sort of wrap up, I want to show you an example of that. So what we did, we took several different samples that would sequence to normal coverage, 30-fold coverage, and we identified the genes that are associated with hypertension. And these are things that have been identified by GWAS over the last decades. And because there's lots and lots of different genes involved and lots of alleles, everybody ends up with a different score, and you can work out what your score is relative to somebody else. And so the sample that I'm going to talk about, which is NA12878, is the red circle. And you can see that that individual scores reasonably highly among this selection. But what we're really interested in doing is seeing if that score that that person gets for hypertension is robust when we use smaller amounts of data. So going from 30x, we wanted to downsample down to 5x, all the way down to 0.5x, and that's shown by the dots on the right-hand side of the graph. And hopefully you can see that they do all fit on a line. So we do have a robust way of identifying these risk scores at low sampling, at low coverage. And we're not just limited to looking at hypertension. We can also look at essentially any complex disease where the associations have been worked out by GWAS. And so here we're showing another 20. And it's a similar sort of thing, that on the left-hand side, we've got all of those samples sequenced to 30x, and on the right-hand side of each of the subfigures, we've got NA12878 downsampled between 0.5x and 5x and repeats. And so hopefully you can see that for each of these, we've got quite a tight distribution. It's quite a robust way of doing things. So I just want to wrap up by saying that this is something that you can do with the reads that you are not enriching for, with your rejected reads from an adaptive sampling run. Your adaptive sampling panel could be anything you want it to be, but in this case we thought that complex disease and PGX panels went together very well. I think that's all I've got time for. If you've got any questions, like I say, be very, very happy to answer them. Please come and talk to either me or Cecil afterwards, and we'll do our best. Thanks very much for your attention.